Hello and welcome to this webinar on the role of flocculation in the recovery and purification of monoclonal antibodies. My name is Des O'Grady. I'm the Market Development Manager for Mettler Toledo's In-Situ Particle Characterization Group. So I'll quickly take you through the agenda for the webinar. Firstly, we'll look at the role of flocculation itself in cell suspension separations. We'll look at how to effectively track changes in floc count and size and how to track the flocculation process itself, specifically in situ and in real time. Thirdly, we'll look at some case studies focused on, first of all, screening flocculants for their effectiveness, correlating floc size and flocculation performance with filtration rates, and then look at studying the impact of process parameters on flocculation. Finally, we'll wrap up with some conclusions and takeaways. So here we have a pretty nice process flow diagram that illustrates nicely the recovery and purification of monoclonal antibodies, specifically the steps involved moving from harvesting all the way through to ultrafiltration and diafiltration. So you can see there are a number of steps along the way through this recovery and purification process, but really where we're going to focus for today's webinar is right at the start. This is the harvesting stage, essentially where we're looking at the removal of cells and cell debris prior to the first chromatography stage or first chromatography operation. Essentially this step, like I said, involves the removal of cells and cell debris from the culture broth and then clarification of the cell culture supernatant, which actually contains the antibody product itself. One thing to consider and is happening in the industry is that cell densities are increasing in bioreactors, typically in order to achieve higher product titers. But along with this increase in cell density, problems, well-documented problems begin to appear. So these include decreases in cell viability, potentially increase in impurity levels moving through the supernatant, broader particle size distribution, which can cause separation and clarification issues, and then, like I said, ultimately an inability to separate the cell suspension itself, or if not an inability, certainly a lot of difficulty surrounding that separation step. Typical approaches to the separation step involve disc stack continuous centrifugation coupled with depth filtration. At elevated cell densities, there can be a burden placed on the filtration train and it can be difficult to effectively separate cells and cell debris. And often the cost of separation increases with the depth filter surface area requirement. So as cell densities increase, typically this depth filtration step becomes more difficult, more expensive, and the cost of separation can sometimes become prohibitive or at least limit the return on investment associated with the step itself. One technique that's emerged in order to help with this process is flocculation. So it can be an effective pre-treatment where the cells and cell debris are actually treated with a flocculating agent in order to increase their size, reduce the number of fines present, and ultimately facilitate an effective separation process. And this simple picture here just shows what a typical flocculation process looks like. This is actually the flocculation of pond water, typical common process where algae is removed through flocculation. So you can see that picture on the left, we have a large number of small algae particles. They're flocculated together to form a small number of big particles, and essentially this clarifies the solution. Some of the pros and cons of flocculation for the separation of cells and cell debris are as follows. So the pros are obviously it can help improve the clarification efficiency and the separation efficiency. It's possible to achieve higher recovery yields, potentially through using flocculation. It can help improve depth filter capacity, and it can facilitate high cell density separations. However, there are cons associated with using a flocculating agent as well. There can be an impact on downstream chromatography operations, especially if this, typically a polymer, ends up moving through with the supernatant to the chromatography step. Sometimes, or almost always, there's a need for downstream removal of the flocculant to acceptable levels, and there's guidelines that the FDA provide around this. And then finally, there's an additional cost associated with any pre-treatment step. It's essentially another unit operation, and there's a cost associated with the flocculant itself. So taking into account these pros and cons, if the choice is made to move forward with flocculation, it's certainly important or even critical to characterize the flocculation process effectively in order to develop and scale this pre-treatment step effectively. So what we're going to talk about today are some tools and techniques available to scientists engaged in this flocculation step and the optimization of the step itself. So just some background on fluid particle separations. Typically, particle number, particle dimension, and also particle shape all play a role in filtration and centrifugation. And taking it to a very simple degree, the image on the left indicates something that's difficult to filter, where we've some big particles but also a lot of fine particles, and the image on the right is obviously easier to filter. This is simply because these small particles block the channels and the pores created by the larger particles and make it very difficult for the centrate to move through the filter to the outlet. For a flocculation pre-treatment, it's critical to consider a couple of items. So firstly, fines capture is very important. So how many of those very fine cells or the cell debris can we capture and bring together to form larger flocs, since fine particles play a critical role in the efficiency of this filtration or centrifugation step. Secondly is the mean floc size. So larger flocs separate more effectively. And also the uniformity of the floc size helps improve the separation efficiency also. Finally, the consistency of the flocculation from batch to batch can be important. So essentially in any bioprocess, the raw material can be consistently changing. So it's very important to ensure that the flocculation process itself can adapt or be robust to changes in the raw material feed coming through. So the cell culture broth coming through, the flocculation step might have to adapt to differences in cell density, for example, in order to facilitate a consistent separation using the equipment that's available. So now we'll look at some tools and techniques available for effectively tracking the flocculation. So some challenges associated with sampling floc systems include the propensity for flocs to break up when you sample. Flocs, by their kind of very nature, are quite delicate. So as you sample, as you prepare the sample for measurement, and even as you measure the sample, there can be the potential to break apart flocs, leading to misleading results or potentially erroneous data. Secondly, there can typically be a long time delay between sampling and delivery of results. Sometimes this is acceptable, but a lot of times, especially for fast-changing processes, it's necessary to get more real-time information or real-time feedback about how the flocs or the flocculation process is changing. Thirdly, many samples need to be taken in order to establish the flocculation kinetics. So typically a sample might be taken at the beginning of a process or at the end of the process in order to understand the beginning state and the end state. But in order to establish kinetics and to understand or to develop a deeper understanding of this flocculation process, many samples need to be taken, and this can be a limitation. When there are fast process changes, which can be typical in flocculation, it's possible to miss these changes if you rely on sampling. So you might take a sample at just the wrong moment and may miss the flocculation process itself. So these are some of the challenges associated with sampling floc systems. And this example is just a kind of a nice example that shows some of the limitations associated with sampling. So here we are actually looking at crystals, which are much more robust and less delicate than flocs, but you'll see that they can have a propensity themselves to break apart when sampled. So if we look at this crystal system where we get these rod-shaped crystals, when we look at an in-situ microscope image on the left and we compare it to an offline microscope image on the right, you can see that they're pretty similar. So in this case, the sampling has been pretty representative. The in-situ image matches the offline image quite nicely. However, in this case, for a different system, we can see that the sampling really has been non-representative. So the in-situ image on the left actually shows these very delicate crystal structures which, when you sample them, break apart. And you can see that under the microscope slide, what we see is really a lot of very small, fine material, but in reality, in the system itself, those crystals or those particles have actually been much larger, kind of delicate structures. And this for me is potentially indicative of what might happen a system of flocs when we sample, is that they could break apart, and any measurement that you try and make just may be misleading. So the tool we're going to focus on today in order to help understand and track the flocculation process is particle track with focus beam reflectance measurement technology. So essentially this is a probe-based technique that tracks the rate and degree of change to particles and particle structures as they exist in process. And it's especially suited to tracking flocculation processes because not only does it track changes in particle size, but it also tracks changes in particle count. So it's a very sensitive and precise measurement of how particle size and particle count is changing. And in a flocculation system, what we have is a large number of small particles sticking together essentially to make a small number of big ones. So we have changes in the particle count, and we also have changes in the particle size for a flocculating system. So the tool is very well suited to tracking this. It's also a probe-based technique, so sampling isn't required, so you avoid this sampling issue. We actually make the measurement directly in the process itself, and essentially for flocculating systems, it can be a very useful tool to track changes and understand what's happening in there. So here we're actually going to look at how the FBRM probe tracks a process that's flocculating. So the fundamental measurement of the FBRM probe is a chord length. And a chord length is simply a measurement of a particle that's related to the particle size. And what we do is build up a distribution of these chord lengths that are associated with the number or the count of chord lengths measured. So if we look at the distributions that I'm showing here, these are simply 3 chord length distributions measured at 3 points in our process. And on the x-axis here is this chord length related to the particle dimension or the particle size. And on the y-axis is the particle count. So really these chord length distributions are sensitive to changes in the particle size or dimension, and they're also sensitive to changes in the particle count. So you can see the blue distribution, we have a very large number of particles around 10 microns, and this is at 0 minutes. And then after 90 minutes, you can see that the number of smaller particles around 10 microns has decreased, and we're seeing a subtle shift to the right in the distribution. So the overall particle size, or chord length in this case, is increasing. At 180 minutes, you can see this process is even more pronounced. You can see you have much less small particles, and we're seeing more and more of those large particles around 100 microns. And this is classic data that tracks a flocculation process. So we're seeing a decrease in the number of small particles. These small particles are sticking together to form larger flocs, and we're seeing an increase in the number of larger flocs. And overall, we're seeing a shift in the distribution towards the right. So an increase in the chord length and an overall increase in the size of the particles or the flocs that we're measuring. So one nice thing that you can do with these chord length distributions, I've only shown 3 here at 3 points in the process, but typically we're measuring a new chord length distribution every few seconds in a process. So what you can do is you can actually isolate statistics associated with counts in individual size classes. So here we're counting all the particles less than 50 microns for every chord length distribution measured. And here we're actually taking a new statistic, which is the counts between 50 and 1,000 microns for every chord length distribution measured. And then we trend these over time. So if the chord length distribution itself is a fingerprint or an indication of the particle size and count at a specific point in time, these statistics, which are trended over time, really give us an indication of what's happening throughout the whole process. So for this set of data, we can see we start off with a very large number of small counts in red, and they gradually decrease over time. And as they decrease, you can see an increase in the number of larger counts between 50 and 1,000 microns. And then eventually, towards the end of the process, we reach a steady state. So these are the 2 ways of looking at FBRM data. The first is the chord length distribution itself, and then we can take statistics from the chord length distribution and trend them over time. So here we can begin to understand what's valuable about this data. So the first piece of information we get from these trended statistics is onset. When does something start? So for a flocculation process, it can be very useful to understand what concentration or what dosage of flocculant is required in order to make the flocculation process begin. And that can be valuable because you don't want to overdose expensive flocculant. So you just want to dose enough maybe to start the flocculation process. The second thing is the rate. So here we can see very clearly the rate of change. How fast is something changing? And the rate of a flocculation process can often be associated with this strength and stability of the flocs formed. So this can be very useful information to acquire also. The third piece of information is the endpoint. So when does this process stop? Is there a certain concentration or dosage of flocculant that can be added where no more flocculation occurs, which will be a waste? So we can really see endpoint using this technique. And then the fourth piece of information is the degree of change. How much bigger or smaller did our flocs get? How effective was this addition of the flocculant? Did it make really big flocs? Did it have no impact at all? Was it somewhere in the middle? So these four pieces of information can be very critical for understanding and characterizing a flocculation process. That's onset, rate, endpoint, and degree of change. Finally, the trend I showed in the top right-hand corner is really focused around counts in individual size classes. We can also obviously take something like the mean cord length, which is simply the mean of the cord length distribution, and we can trend this over time as well. So this gives us a more broad indication of the average floc size and how that might be changing over time also. So we'll move on to some case studies where the use of the tools or the use of the FBRM technique is actually shown for some systems that flocculate. So here we're looking at classic FBRM data associated with a process that's flocculating. So we're looking at how three statistics are changing over time. And the statistics that we've chosen are the counts less than 50 microns, so the small material in our system, counts between 250 and 1,000, which is obviously the larger material in our system, and then finally the mean length weight, which is simply an indication of the overall or average floc size. So what we can see is at the start of our process, between 0 and 10 minutes, the number of particles between 0 and 50 microns, the smallest particles, is quite high. And the number of particles between 250 and 1,000 is essentially 0. So you can see that purple trend is trending very high, but that orange trend is trending very low. And the overall floc size, this mean length weight, is quite low as well. Now we can see the exact point when our flocculant is added. And right at this point, you can see a drastic decrease in the number of fines and the number of counts less than 50 microns. And we can see a dramatic increase in the counts between 250 and 1,000, and also the mean floc size or the mean cord length. Now, the next thing that happens is that all of this has occurred at 300 RPM. Now, you can see the point where the agitation in this system is increased to 800 RPM at around 15 minutes. And as soon as you increase to 800 RPM, you can see the reverse process happens. You can see that the mean floc size, you can see the counts between 250 and 1,000 decrease dramatically and the number of fines increase. So essentially what we're doing here is as we increase the mixing rate, we introduce shear into the system and everything breaks apart. And then the mixing rate is reduced back to 300 and we can see there's some degree of reflocculation. So right around 20 minutes there, we can see that the system begins to reflocculate with the fines decreasing and the mean increasing again. So this is a classic experiment that shows the influence of adding the flocculant on our system and also the influence of shear. And this can be a very useful system to experiment to understand how changing shear conditions affect our flocculant process, and it also helps us understand the strength of the flocs that we're making. So do they get significantly destroyed? Do they reflocculate as we change shear in this process? So there's really a lot of information contained in a single experiment, and this count-based and size-based information and how it's changing over time can be very useful to help understand what's happening in a flocculating system. Taking this experiment, we can look at the influence of the actual type of flocculant we use on our system. So here we're looking at 10 experiments where 10 different flocculants were used, and we're looking at the effect of the flocculant type on the counts less than 50 microns for the experiment that I just described. So this really becomes a very effective screening tool for understanding what impact different types of flocculant have on our system. And it's a very fast way to characterize or screen these flocculants for effectiveness. Here we're looking at the counts less than 50 microns. And here we're looking at the mean cord length trended over time, again for these 10 different flocculants. So very quickly we have this ability to understand the influence of the flocculant type on not only the fines capture, but also on the average floc size that we're measuring and how that changes under different shear conditions. Now the reason I bring this up is that there's a very nice reference available, and this is also a standard technique that's used in a lot of different places, where you can actually use this information gathered with the FBRM to understand the time for deflocculation and the time for reflocculation. So basically these are equations that can be developed that characterize how simple it is to deflocculate a system under increasing shear conditions and how much, or the propensity for a system to reflocculate as you reduce the shear level or reduce the mixing rate again. So these exponential functions, where we can see these time functions TDF and TRF, are essentially a time to deflocculate and a time to reflocculate. And if we look on this piece of info, or this slide, we can see that we can really begin to characterize what effect the flocculants have on the flocs themselves. So a high time for deflocculation indicates that they have stronger flocs under shear conditions. And then a higher TRF indicates that the flocs won't reflocculate after a shear event. So this gives very nice information about the influence of our flocculant on its ability to produce strong flocs that are stable. And from a separations process in a centrifuge, it's important to generate strong flocs that won't break apart under shear conditions as they move through pipelines, as they move from tank to tank, and as they move on to the filter or the centrifuge itself. So you could go through all this work where you actually flocculate your cell debris, for example, only to have it deflocculate under varying shear conditions before it gets to the centrifuge itself, and that would defeat the whole purpose. So this is a very nice experiment to understand the stability and the strength of the flocs that you're making using different flocculating agents. So now we'll look at the impact of floc size on filtration rate. So this is actually very nice work from Amgen that was presented at an AICHE meeting in 2010, and I have the reference below where you can actually get these slides that Anna Senzuk presented. This is a really nice paper, and I'd recommend that people look at it. So here they're actually looking at the flocculation of cell debris, and we can see with this data just the classic example, just like I showed before, of this flocculation process. So it's a certain concentration of a certain type of flocculant, and you can see that the smaller particles, this blue line at the top, the counts between 0 and 10 microns, decrease over time between 0 and 30 minutes. And if we look at the larger counts between 50 and 100 microns in green, you can see they increase over time. So this is a very simple example showing the flocculation of cells and cell debris in a culture broth. Here we can actually look at cord length distributions trended over time, or different cord length distributions taken at different times in the process. So here we begin to see the decrease in the counts of the cells around 10, 20 microns, and an increase in the number of counts above 20 microns. So here you begin to really see the effect of this flocculation process as we get larger and larger flocs forming over time in this process. Finally, the final floc size can be correlated to the separation efficiency. So in this case, we're looking at settling Vmax. Vmax is an indication of separation efficiency. The higher the Vmax, the faster the separation. And you can see that as the mean particle dimension measured with the FBRM increases, we get a faster and faster separation rate. So this is nice modeling work that was done that was used to help predict filtration performance and also to help pick the conditions of flocculation in order to target a specific floc size that would give the most optimal separation efficiency. So I'd highly recommend, if you're interested, to check out this presentation. It's available online at the link below to learn more about this process itself. So next we'll look at the impact of process parameters on flocculation. So in this example, we're actually going to look at the flocculation of yeast cells. So here we have 3 examples of 3 different process parameters: temperature, pH, and the concentration of the magnesium cation. And we're looking at the effect of each of these parameters on floc size. So D in each of these cases, the variable D, is the mean cord length measured with the FBRM. And this reference here, you can go into a lot more detail on the experimental setup and the work that was done for these examples. But what we can see is that if we look at the top left, the effect of temperature on the floc size measured with FBRM. So we can see a clear drop in the floc size as temperature increases, especially around 60 degrees here, where we see this pretty precipitous drop in the floc size. So clearly temperature plays a role in the stability of this floc. These flocs break apart as temperature increases. The second example is pH, where we see a much more stable relationship between pH and the mean cord length of the flocs measured. Finally, we can see a relationship between the concentration of the magnesium cation and the mean cord length of these flocs measured. As the magnesium cation concentration increases, the floc size actually increases as well. So the FBRM technique is a very nice way to understand the effect of various process parameters typically found in a bioprocess, for example, on the floc size or the size of these flocculated material. So let's wrap up with some conclusions. So what we've covered today really shows that flocculation can be a valuable pre-treatment to support the effective separation and clarification of cell culture fluids. We know that the characterization of the rate and degree of change to floc size and count is critical for effective process development and characterization. Sampling of flocs can be very difficult due to their delicate nature and the need for more representative sampling methods. And finally, I hope it's been shown, even in a brief way, that the particle track instrument allows scientists to track the evolution of floc size and fines capture over time, hopefully screen the most effective flocculants for a given application, target a certain floc size that will improve separation and clarification efficiency, and then ultimately build process understanding around these difficult to characterize flocculation processes. I'd like to thank you for your time. Thanks very much.